I'm your host, William Bra. I am the Executive Director of the Venture Development Center and Assistant Vice Provost of Research at the University of Massachusetts in Boston. Today we're going to be talking about the Gulf oil spill, using modern-day biology to assess the environmental impact and to help in remediation. I'm going to be moderating this panel today with my co-moderator, John Sterling.
Thank you, Bill. I'm John Sterling, Editor-in-Chief of Genetic Engineering and Biotechnology News. Genetic Engineering News is sort of the longest running biotech publication completely focused on biotech. Why I'm here today is this whole issue of the Gulf oil problem, not just from a societal and economic point of view, but obviously from a scientific point of view. The ramifications were astounding as we all watched what happened. The title of our discussion today is The Global Effect of the Oil Spill. The great news is today we have an expert panel in environmental and biological sciences.
I have been doing research on the biogeochemistry, that is how compounds such as petroleum, hydrocarbons move through the environment, how they interact with the biota in the system, and their intermediate and ultimate fates. And I've been involved in oil pollution research for 30-some years now.
I'm a postdoctoral fellow at Lawrence Berkeley National Lab, and I'm really interested in looking at how microbial communities respond to perturbations in the environment. And so I'm specifically doing research on how the deep sea microbial communities were affected by the oil spill.
I am a professor of marine microbiology at Scripps Institution of Oceanography, and I'm interested in the genetics and the genomics of the adaptations of deep sea microbes. And in this discussion, I'm very interested in the influence of oil infusion into deep sea communities and their influences on the deep sea microbes.
Pressure Biosciences is involved in developing products, tools that can be used by researchers around the country, around the world, to aid them in their research. We base these tools, these products, on something called pressure cycling technology. Our goal, our mission as a company, is to work with researchers like Olivia and others to try to develop the tools that can enable them to use the science that they have to the betterment of everyone.
My research interest is really focused on zooplankton ecology and looking at the distribution of zooplankton. Plankton are the small animals, plants and animals in the ocean, the base of the food webs. And what I'm really interested in, or do a lot of my research, is these interactions between the different plankton communities and the feedbacks that go between zooplankton and bacteria.
Winnie, let's start out with your work, specialty in zooplankton ecology. And before we get to the broad implications and the effects of what that oil spill actually did, let's find a little bit more about how the intricacies behind zooplankton biology.
So the base of the marine food web in the ocean, from the very plants, the phytoplankton that are there in the ocean, these small single-celled plants, their distribution controls the further up the food web, so the zooplankton that I'm interested in, which all of these things are then the food for the commercial fish that are there and for the higher up in the trophic levels for marine mammals and whales.
How are zooplankton related to bacteria? What are the interactions?
So zooplankton, these are small little animals that drift in the currents. And as they're feeding on these phytoplankton, a lot of them are real messy eaters, and they dribble their food out, just like kids do. And by doing this, they're putting a lot of organic matter into the water column that feed the bacteria and help the bacteria to grow.
I'm curious how changes in the bacterial community composition will affect the interactions between other plankton groups and nutrients.
That's one of the big areas of research right now, and an area that we don't know, and a potential area of a lot of interest with oil spills. And also, if the bacterial community is changing or responding to the perturbations, what does that mean further up trophic webs?
What's being done to learn more about what happened in the Gulf and how we might deal with a future oil spill, which I'm sure sooner or later is going to come?
One of the areas of research and questions is the chronic effects. So what's the long-term effect up the food web, and then what's the impact to fisheries and other commercially important biota in the system. There might not have been an immediate response to the BP spill and the oil spill in the first weeks or months, but it might be months, years, 2 years down the line that we see the effect.
I have a question for Dr. Farrington. And in particular, I'd like to know if the bacteria will degrade the oil or in the hydrocarbons in the column, water column.
One thing that's very clear is that from studying various aspects of oil pollution in the marine environment, this is the BP spill in the Gulf. That's only the latest in a number of different spills that have occurred, not just in the United States, but all over the world. The big problem is that we do not, or up until recently, we've not had the tools to really be able to look at degradation of oil actually in the environment other than to look at the chemistry. Now we do have the tools, as been pointed out from Olivia's lab group and others, to take a look at what microorganisms are actually there. My final comment is that in the Gulf area, we've already had a number of natural seeps going on for a long period of time, and so there's been an evolution of microorganisms in association with those seeps, as well as higher parts of the food web in those specific areas. In addition to natural seeps, there have been chronic inputs from land for a long period of time, and also during Hurricane Katrina, there were spills of oil as a result of damage to oil facilities on land.
It seems to me that you're saying that there's a natural process that's taking place, although we've introduced a specific mix of compounds in the oil.
Correct.
But wouldn't that just kind of over time take care of itself?
There are areas where we have looked at oil spills for long periods of time. In fact, there's one only an hour and a half away from here on Cape Cod where they had an oil spill in 1969 that has been studied. Just recently, a few years ago, people revisited one of the marsh areas that was very heavily oiled. And the oil is still there in the sediments. If you look at the other areas of the marsh, it's luxuriant, it looks fine. And so it depends on where you are. And that particular oil spill, by the way, was probably 1/100th or 1,000th of the total spill in the Gulf. So you'll find areas in the Gulf where there will be impacts for as long as a year, if sometimes a decade, and there will be areas that are recovering right now.
One issue that I wanted to pick up on, John, is the fact that oil is natural. And I think that's a really important point to make. Microbes have been evolving for millions of years to deal with oil.
I would just like to comment that I have no disagreement with either of those comments, with the caveat that mercury is also a natural chemical, and so is arsenic. And the issue at stake is how we as a society mobilize these chemicals, put them in slightly different forms, and move them around and introduce them in rates to a particular area that are not natural.
The question really that we're trying to get at today is how close do we need to look about a sudden shock to the system and its ability to cleanse itself over time.
There was a bloom of what are called gamma proteobacteria. We found a group of bacteria called ocean spirales, and we find those in the uncontaminated sites as well.
Mm-hmm.
So these bacteria that are there that appear to be capable of degrading oil.
I think Olivia's study shows how the modern tools of genomics, for example, can be brought to bear on the question of who's there, what's their metabolic potential, and how can that be harnessed?
We're coming at it from a different point of view. We're coming at it from a industrial point of view. The tools now are so much different than they were 10 and 20 years ago. There has to be cooperation and collaboration between industry and academia and government. We need the funding, we need the support of government, and we need the support of academia to let us know what do you need for tools so that we in industry can put our sights on developing that next generation of tools.
There's a long way to go, but I do agree that the tools aspect of it has been a huge advantage.
Now with the tools that we have today, I think we need to think of the environmental context of any oil spill, the sorts of nutrients that are there for the microbes to utilize, not just their ability to break down the oil, but their ability to get the other nutrients that they need.
In the research that I'm doing, we don't have the identity of the organism and a link with its genes that allow it to break down oil. We'd like to really piece together large segments of DNA where we can get the identity of the organism as well as the functional genes that allow it to do things like eat oil. And so we're actually using pressure for that reason. Although we have a lot of molecular tools now, it did pose a challenge because we just couldn't get enough DNA, because you can imagine how precious these samples are and you get very little material to work with. So we've been able to introduce pressure cycling into our extraction protocols and have gotten a lot more DNA that have allowed us to do something like metagenomics.